Hello students, in this video we are going to discuss about a very important topic from the chapter Molecular Basis of Inheritance, that is translation. So what is mean by translation? So already we have discussed about the transcription. So this is the continuation of the same topic. Transcription is followed by translation. So translation is actually the second step involved in the protein synthesis or in the gene expression. So translation refers to the process of polymerization, of amino acids, process of polymerization of amino acids to produce a polypeptide. So polypeptide is nothing but protein. So what happens during this translation actually? So this is a one amino acid. The amino acid is linked to another amino acid. Again, this second amino acid is linked to one more, third amino acid. So like this, many amino acids are linked together to form a chain of amino acids. So the process of formation of amino acids, chain of amino acids, so that is called as polymerization, and which leads to the production of a protein is known as translation. So which cell organelle is responsible for this translation? Already we have studied about this particular cell organelle in first PUC, that is ribosome. So ribosome is called as cellular factory for the production of proteins, or responsible for the synthesis of proteins. And you know, the ribosomes are the most abundant cell organelle in the cells. So this is the most abundant cell organelle in the cells. And the ribosomes are basically known as nucleoproteins. These are basically known as nucleoproteins. And you know, the ribosomes are made up of two subunits. So this is a smaller subunit and this is a larger subunit. This is a larger subunit and this is the smaller subunit. So this is the definition of translation. Now let us move to the next important part of this topic, that is the steps involved in the translation. Actually, 4 important steps are involved in the translation. See, the first step is charging of tRNA, or it is also called as amino acylation of tRNA. So just remember, if it is the cell, eukaryotic cell, if it is a eukaryotic cell, within the eukaryotic cell there is a nucleus, and the nucleus consists of DNA. So these are the DNA. And during the transcription, mRNA is synthesized as well as the other types of RNA synthesized. And through the nuclear pore, these mRNA will be released into the cytoplasm. So these are the nucleotides found in mRNA. This is a 5' end and this is a 3'. And in the cytoplasm, these ribosomal subunits are also found. Just remember, the ribosomal subunits will associate, or they will come together only at the time of translation. Till then they will found in the form of dissociated form. So now, according to the information present on this mRNA, there is a need to synthesize the proteins. But for the production of proteins, amino acids are required. So these are the amino acids. And these amino acids are found in the cytoplasm. And these amino acids are found in the cytoplasm as a neutral molecule. So the first step is charging of such amino acids, and then linkage of such amino acids, charged tRNA to the tRNA, because tRNA is the molecular vehicle. This tRNA will carry the amino acids from the amino acid pool to the mRNA. Now, according to the information present on mRNA, the proteins will be synthesized by the help of these ribosomes. For that, amino acids are required, and these amino acids are carried by the tRNA. So this we will discuss in this steps. Now, first step is charging of tRNA or amino acylation of tRNA. So these neutral amino acids now undergoes charging. That means amino acid plus ATP. ATP is the energy currency. So it will provide energy to the amino acid, and it leads to the formation of amino acyl AMP. It leads to the production of amino acyl AMP. And in the next step, so this is the first step. In the second step, the amino acyl AMP, amino acyl AMP means the charged amino acid that reacts with the tRNA that acts as a vehicle, molecular vehicle, and it leads to the formation of amino acyl AMP. Amino acyl. It leads to the formation of amino acyl tRNA. Amino acyl tRNA. So this is released. See, this is a required product of first step, that is amino acyl tRNA. Amino acyl tRNA means, say if it is the tRNA, actually tRNA is looks like a double stranded molecule. It is actually found like this. So if it is the tRNA, see here, this is one arm, and this is the another arm, one more arm, like this. So this is the structure of tRNA, and here it consists of 5' end, and this is a 3' end, and here there is a particular site called as amino acyl binding site, and now to this particular site there is an attachment of amino acid. So this charged amino acid is linked to the 3' end or third prime end of the tRNA, and this structure is called as amino acyl tRNA. Amino acyl tRNA. So this is the first step in translation, and this process is carried out by the help of an enzyme that is called as amino acyl tRNA synthetase enzyme. Amino acyl tRNA synthetase enzyme. So this is regarding the first step involved in translation. So it involves, in this phase, the amino acids are activated in the presence of ATP and linked to their cognate tRNA. Cognate means the recognizing tRNA. So each amino acid is carried by a specific tRNA. So that is called as cognate tRNA. Amino acyl tRNA synthetase enzyme is involved in this process. Now let us move to the next step, that is chain initiation. So what is this chain initiation? What happens during chain initiation? So in the first step, activation of amino acid takes place, and such activated amino acids are linked to the tRNA, and such amino acyl tRNA are now used for the synthesis of a polypeptide chain. Now just see here, see this is the mRNA. This is the mRNA. And the mRNA is made up of nucleotides. So these are the nucleotides. Okay. And this is a 5' end. And this is a 3' end. And the nucleotides, the sequence of nucleotides, that depends on the mRNA. It varies from mRNA to mRNA depending on the genetic information present in the DNA. For example, for G, C, C, A, U, G, U, C, U, G, U, A, G, like this. One sequence is there. As I told earlier, the sequence, it varies from RNA to RNA. Let us imagine this particular sequence is found in one particular mRNA. Now, how it undergoes translation? So translation is the synthesis of a protein according to the information present on this mRNA. And the cellular factory responsible for the synthesis of protein is ribosomes. Now, during the chain initiation, in the first step, the smaller subunit of ribosome will bind to the mRNA in such a way that the first triplet codon is always initiator codon. Which is the initiator codon? That is AUG. But it is present not at the beginning. Yes, of course, it will be there at the beginning of the coding sequence. That means the entire mRNA does not code for amino acids. So at the 5' end as well as the 3' end, some non-coding sequences are also there. The coding sequence begins with the initiator codon, that is AUG. See here, this is the AUG, and it ends with a terminator codon, that is in this case that is UAA. Now the smaller subunit of ribosome binds to the mRNA in such a way that it covers two triplet codons. And this first triplet codon is AUG, that is the initiator codon. It is followed by the binding of amino acyl tRNA. It is followed by the binding of amino acyl tRNA. Amino acyl tRNA means amino acid with the tRNA. So this particular amino acyl tRNA consists an anticodon, that is UAC. See here, A is complementary to U, U complementary to A, G complementary to C. So codon and anticodons are complementary to each other. That's why a temporary bonding takes place. Then what is the amino acid found in this particular amino acyl tRNA? It is always a formyl methionine, because initiator codon, that is AUG, codes for formyl methionine. Now it is followed by the binding of larger subunit of ribosome. The larger subunit of ribosome binds to it. So now the initiation complex is completed. But just remember, in this larger subunit we can see two sites. This is the one site and this is another site. And this site is called as the P site and this is the A site. See here, the P site is filled with the first amino acyl tRNA. First amino acyl tRNA. So consisting an amino acid, that is formyl methionine. But A site is empty. So this is a larger subunit of ribosome. This is a smaller subunit of ribosome. This is a site, P site, and this is a initiator tRNA with the amino acid formyl methionine. And this is a mRNA. So this is regarding the structure of initiation complex during the chain initiation of transcription. Okay, now, so for initiation, the ribosomes bind to the mRNA at the start codon, that is AUG, that is recognized only by the initiator tRNA consisting amino acid formyl methionine. In inactive state, ribosome exists as two subunits. So when there is no translation, when there is no protein synthesis, the ribosomal subunits will be found in the dissociated manner, and they will join together only at the time of translation. A large subunit and smaller subunit will be found separately when there is no translation, but during translation they will join together. When the smaller subunit encounters an mRNA, the process of translation of the mRNA to proteins begins. Now, the next step is chain elongation. See, again we need to draw the same diagram. There is a mRNA. So this is the mRNA consisting nucleotides. So we have written AUG here, AUG, followed by UUU, CUG, UAA, and it will continue, as well as it is preceded by, AUG is preceded by some nucleotides, and this is a 5' N and this is a 3' N. So already the initiation has begun. Initiation is begun. Now the elongation takes place. See, this is the smaller subunit of ribosome, and this is the larger subunit of ribosome. As I told earlier, here the first amino acyl tRNA is found, and that consists amino acid, that is formyl methionine. So this is the structure of initiation complex, and here UAC anticodon. So, and the first site, this is a P site, and this is the A site, and the A site is empty. So during chain elongation, this A site is recognized by another amino acyl tRNA consisting a specific amino acid. So why this particular tRNA binds to this mRNA or this codon at this particular A site? Because it consists an anticodon AAA. You know, A is complementary to U, so it will bind to the codon UUU in the A site. And this amino acyl tRNA consists of specific amino acid, that is phenylalanine. So this phenylalanine is recognized by the codon UUU. So this is about the genetic coding system. Now, after the arrival of amino acyl tRNA to the A site, there will be the formation of a bond between the amino acid found in P site and A site. So, the bond formed between the amino acids found in P site and A site is called as peptide bond. So, this bond is peptide bond. This bond is the peptide bond. When there is a formation of peptide bond between the amino acids found in P site and A site, the bond present between the first amino acid and the first tRNA will break. So, this bond will break. So, this tRNA becomes empty. This tRNA becomes empty. But the second tRNA consists 2 amino acids. Now, it is followed by the movement of a smaller subunit one step forward. It is followed by the movement of ribosomal subunit one step forward. And one more very important thing you should remember, this bond is the peptide bond. This bond is peptide bond. And this bond is formed by the help of an enzyme called as peptidyl transferase enzyme. The name of that bond is peptidyl transferase. So, peptidyl transferase enzyme is responsible for the formation of peptide bond. So, now the smaller subunit moves one step forward. The smaller subunit of ribosome moves one step forward, and it is followed by the movement of larger subunit of ribosome. So, when the smaller subunit moves one step forward, it is followed by the movement of larger subunit of ribosome. So, now this is the empty tRNA. This empty tRNA will be removed, and this empty tRNA will bring another amino acid if it is available. Okay, just see here. Now, this tRNA is shifted from A site to P site. Now, this is a P site and this is a A site. So, the amino cell tRNA is shifted from A site to P site. So, again A site becomes empty. And one more thing, this tRNA consists of 2 amino acids. 2 amino acids. Now, during elongation, this empty A site consists of a codon, that is CUG. It is recognized by one more amino acyl tRNA. One more amino acyl tRNA with a specific amino acid. See, the codon present here is anticodon present in this tRNA is GAC, because codon is CUG. And this tRNA consists of a specific amino acid, that is leucine, because leucine is coded by CUG. And now it is followed by the formation of peptide bond. Peptide bond between amino acids found in P site and A site takes place by the same enzyme, that is peptidyl transferase. Particularly in prokaryotes, this is 23S rRNA. So here this is not the proteinaceous enzyme, this is a ribozyme. RNA acts as an enzyme. And it leads to the formation of breakdown of this bond present between the amino acid and tRNA. And again, now it is followed by the movement of smaller subunit one step forward, followed by the movement of larger subunit one step forward. The larger subunit is also moved. So, now the removal of or releasing of this tRNA. Okay. So, now this tRNA consists of 3 amino acids. So, this amino acid is coded by CUG, phenyl coded, alanine coded by UUU, and AUG codes for formyl methionine. So, the last codon is arine, and this is a terminator codon. This I will explain in the next step, that is during the chain termination. Now, let us see the important points from this topic. This step, that is chain elongation. There are 2 sites in the larger subunit for subsequent amino acids to bind to and thus be close enough to each other for the formation of peptide bond. The ribosome also acts as a catalyst. 23S rRNA in the bacteria is the enzyme, that is also called as ribozyme, for the formation of peptide bond. During this stage, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the RNA anticodon. The ribosome moves from codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequences dictated by DNA and represented by mRNA. Now, we are moving to the next step, that is chain termination. Again, we need to draw the diagram here to explain. So this is the mRNA, and these are the nucleotides. 5' end, 3' end, AUG followed by UUU, CUG, and then we have written UAA, and some other nucleotides will be there. So till here, coding region from here to here. Okay, now already the smaller subunit of ribosomes or the ribosomes have reached this particular site, that is the coding sequence. So UAA, okay, it was followed by the movement of larger subunit, and one amino acyl tRNA is found here, consisting in a chain of amino acids, and it was leucine, is leucine, and before that phenylalanine, and before that formylmethionine. Okay, now the ribosomal subunits has reached the terminator codons. Terminator codons do not code for any amino acid. You know, this is a P site and this is the A site. A site is empty, but in the A site there is a terminator codon or the stop codons. These stop codons do not code for any amino acid. So instead of amino acyl tRNA, there will be the binding of release factor RF. So this is a release factor. When the ribosomal subunits or the ribosome reaches the terminator codon, the terminator codon is recognized by the release factor. The binding of release factor to the A site or to the terminator codon causes dissociation of ribosomal subunits. So these ribosomal subunits will be removed from this mRNA. So when the ribosomal subunits reaches the terminator codon, these ribosomal subunits will be removed from the mRNA. So these ribosomal subunits are removed. So at the same time, see, the reason for the removal of these ribosomal subunits is release factor. So this release factor causes the dissociation of ribosomal subunits, so that there will be the removal of this tRNA from this mRNA. So from mRNA, the tRNA with the chain of amino acids are also removed. So now the remaining is only the chain of amino acids along with the last tRNA, Now one more enzyme will come, that is peptidyl synthetase. The peptidyl synthetase removes this tRNA from the chain of amino acids, so that there will be the formation of a chain of amino acids. Here we have written very few, only 3 codons, sense codons, so that only 3 amino acids are linked. But the number of amino acid varies from protein to protein. 20 types of amino acids will be there in the protein molecules, and the number, it may be hundreds or thousands or millions. So that depends on the proteins. So this is regarding the chain termination process. So at the end of the process, or at the end of the translation, a release factor binds the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. And this is a diagram. Only one diagram is given in the textbook. You need to draw this particular diagram. So just try to draw the same diagram in your notes and even for the exam. And this diagram actually shows the chain elongation process. Chain initiation and chain termination is not represented by this diagram, but you need to write all these points to explain the translation. So this is regarding the very important topic in molecular biology, or in the molecular base of inheritance chapter, that is translation. Okay, dear students, the remaining topics will be discussed in the next video. Thank you.